After that, perform pre-selective PCR amplification to amplify specific subsets of digested DNA fragments, using combination of selective primers at 3 ends, which are adenosine nucleotide to EcoR1 adaptor and cytosine nucleotide to MsCl2 adaptor, that complementary to adapters and restriction sites, to select only subset fragments, as mentioned in next slide. This is the component of pre-selective PCR amplification. Include DNA template from restriction ligation and EcoR1 adapter with adenosine nucleotide as primer and MsCl2 adapter with cytosine nucleotide as primer and dNTPs and 10x PCR buffer and MgCl2 and Taq DNA polymerase and deionized water. And this is the reaction of pre-selective PCR amplification, 26 cycles, and after finish, add 100 microliter of sterile water to PCR product. After that, undergo selective PCR amplification by adding 2 nucleotide of adenosine and 1 nucleotide of cytosine to primer at 3 ends, to again select subset fragments. But, be attention please. More selective nucleotide. Detect less polymorphism. So, must only add 3 nucleotide, as mentioned in next slide. This is the component of selective PCR amplification. Include diluted DNA template from pre-selective PCR reaction, and EcoR1 with selective nucleotide, A, C, and MsCl2 with selective nucleotide, A, C, and dNTPs, and 10x PCR buffer, and MgCl2, and Taq DNA polymerase, and deionized water. And this is the selective PCR reaction. Include 2 phases. The first phase involve 12 cycles with decreased temperature by 0.7 degrees Celsius, while the second phase include 23 cycles, and after finish all phases, add 8 microliters Formamide loading buffer to PCR product.